And we see it gives mechanical protection, chemical protection, and also does a little bit of circulation. In terms of mechanical protection, we actually see that the brain is floating within the cerebral spinal fluid. So it gives the brain buoyancy. And it reduces the weight of the brain by about 97%. So brain weighs about 3 pounds, so it's going to reduce the weight of it by allowing it to float, and prevents the brain from crushing under its own weight. It also protects the brain and the spinal cord mechanically by also protecting it from blows, jolts, and other trauma that would otherwise cause the brain and the spinal cord to hit the bony wall, and so and to hurt itself. We also see that it gives chemical protection. Chemical protection helps provide an optimal and chemical environment for neurons. Remember, neurons need a very specific chemical environment for them to work optimally. And then also the cerebrospinal fluid is important in a little bit of circulation. Although the brain has very, very rich blood supply, we see the cerebrospinal fluid also helps to nourish the brain by carrying oxygen, glucose, and other needed chemicals to the neurons and the neuroglia. It also can carry away waste products as well. So the question is, where do we get the cerebrospinal fluid? Well, and what is it? Well, it's sort of a clear, colorless liquid. It's a clear, colorless liquid. And its composition is very similar to blood plasma because it comes from blood plasma. So its ion concentration is a little bit different from blood plasma because, and so it's very similar to blood plasma, but it's a little bit different. It's going to be formed by choroid plexuses. And so what we see hanging from the walls and roof of the ventricles, so for the lateral ventricles, for the third ventricles, for the fourth ventricles, are these little tuffets of capillaries, and these are called choroid plexuses. And these choroid plexuses are going to form cerebrospinal fluid. And if we look at these choroid plexuses histologically, we actually see that here we have our capillary with our endothelial cells, and we see that that's actually lined by a little bit of pia mater. Here's our pia mater, protecting our blood vessel. And then we also have a neuroglial cell that is going to be then lining the pia mater, and those are our ependymal cells. Remember, the ependymal cells are going to line the ventricles, and they have the cilia that are able to help circulate that cerebrospinal fluid, while these ependymal cells are also going to be lining these and be part of these choroid plexuses. So there's very specific what is going to come across this barrier into the cerebrospinal fluid. So we have what's called a blood cerebrospinal barrier, and we're very particular about what comes across because remember, this is going to help to nourish our neurons and so forth, and so we have to have a very particular chemical environment around the neurons associated with the brain. You constantly produce cerebrospinal fluid. If you constantly produce it, that means you got to get rid of it somewhere. So if you're constantly producing it, you got to get rid of it somewhere, otherwise it's going to accumulate, and there's not a lot of space in the brain for it to accumulate. And so we see it reabsorbed into the superior sagittal sinus through arachnoid granulations or villi, which are those finger-like extensions of the arachnoid mater into the superior sagittal sinus. And so what needs to happen is the rate of production of cerebrospinal fluid from the choroid plexuses needs to equal the reabsorption rate. So how much you're making needs to equal how much you're reabsorbing. In an adult, we see that the total CSF is somewhere between 80 milliliters and 150 milliliters in an adult. So total CSF is somewhere between 80 and 150 milliliters in an adult, and it's replaced every 8 hours. It's replaced every 8 hours. Here we see, this is going to be our falx cerebri. We can see this is going to be our superior sagittal sinus. If we bring this down, we can see that superior sagittal sinus, and we can see those extensions of the arachnoid mater into the superior sagittal sinus for the reabsorption of our cerebrospinal fluid. You make it from blood, put it back in the blood. You make it from the blood, put it back into the blood. So where do we see the CSF? Well, the CSF is in the subarachnoid space, and we're producing it in the ventricles of the brain. Well, the ventricles of the brain, remember, arise from expansions of the lumen of the neural tube. We kept that neural tube from the neural, the lumen of the neural tube, which is the neural canal. We kept that. We just enlarged it in certain places. In the telencephalon, that lumen becomes shaped lateral ventricles. And the diencephalon becomes the third ventricle. Even the mesencephalon becomes the cerebral duct. And the metencephalon and myelencephalon, their spaces combine to form the fourth ventricle. And so in the blue here, we're seeing those different spaces. And so here we're going to see the lateral ventricles, the two lateral ventricles here. This is going to be your third ventricle. This is going to be your third ventricle. This is your cerebral aqueduct, and then down to your fourth ventricle. And that fourth ventricle is going to be connected to the central canal of the spinal cord, which was the lumen from the neural tube. Well, if you're constantly producing it, you constantly have to reabsorb it. Well, if you're producing it in the ventricles, and here you can see these little tuffets of capillaries, either in the lateral ventricles, in the third ventricle, and the fourth ventricle, you have to get it to areas like the superior